Special senses are a unique type of somatosensation. The special senses themselves do not traverse any of the sensory pathways within the spinal cord. They use unique pathways along distinct cranial nerves to transmit the sensory information that has been received at unique receptor locations that will respond to unique sensory stimuli. So what are the receptor organs for the special senses? Well, for olfaction, we have the olfactory epithelium. For auditory, we have the cochlea. For the vestibular, we have the semicircular canals. For vision, we have the retina. And for gustation, we have the taste buds of the papilla of the tongue. Each of the special senses will be tied to a distinct area within the cerebral cortex. Olfaction, proprioception, and vestibular will be tied to the parietal and frontal lobes. Vision will be tied to the occipital lobe. The auditory will be tied to the temporal lobe. In which all will eventually get integrated in the general integratory area of the cerebral cortex. And most have a link to the frontal lobe, which are tied to the emotional response that we need in order to link the distinct stimulus to a memory within the hippocampal response to the stimulus itself. We cannot have a true memory if we do not have an emotion tied to it. So let's talk about olfaction and gustation to start with. Olfaction and gustation give us our sense of what we refer to as smell and taste. We have the ability to sense somewhere in the neighborhood of 10,000 to 40,000 distinct odors and or tastes. Within the olfactory epithelium, we have somewhere in the neighborhood of 10 to 100 million different receptors. These are olfactory receptors specific to a distinct chemical class. Within the taste buds, we have what are usually referred to as grouped gustatory receptors, in which case we have 50 distinct gustatory cells within the gustatory receptors. The difference in the number of receptors is based on the difference between having a labeled line response within the olfactory epithelium versus the population coding response that we see with the taste buds. Regardless of what you may have heard in the past, you do not have 5 distinct tastes, nor do you have 5 distinct regions for those tastes. So let's take a look at the olfactory epithelium. The olfactory epithelium consists of a receptor where we have a bipolar neuron exiting through the cribriform plate into the olfactory epithelium. In the olfactory epithelium, we have distinct cells that have projections. These projections are like dendrite spines that enter into the nasal concha. Surrounding these dendrite spines are support cells. The support cells produce a serous mucus. The serous mucus that is produced by the support cells and by the olfactory glandular pit are there in order to trap air molecules as they pass through the nasal concha. Lining the support cells are a series of cilia that help to move the mucus along so that these chemicals, these referred to here as odorant molecules, do not engage with the receptor cells for too long. When the receptor cells are depolarized, they send a signal along the bundle of olfactory receptor axons into the olfactory bulb via perforations within the cribriform plate. It's this interaction within what's referred to as the olfactory bulb that initiates cranial nerve 1, or the olfactory nerve. So let's take a look at how these individual olfactory receptors will initiate their response. What ends up happening is that we have the odorant molecule interact with a specific ligand channel, in this case referred to as the olfactory receptor protein. What this does is initiates a G-linked protein response. This G-linked protein response initiates the conversion of ATP to cyclic AMP. The cyclic AMP then opens a sodium channel, leading to sodium influx. This influx leads to a depolarization moment within the olfactory receptor itself, where if we get enough depolarization to occur, we will trigger an action potential through the specialized receptor cell. You can see this schematic in a video if you follow the link either on Blackboard or by copying this link into your browser. Once we get depolarization to occur within the receptor, this depolarization will then be transmitted to the olfactory bulb. From the olfactory bulb, it will then be transmitted along cranial nerve 1. From there, it will go to two distinct places. Here shown as going to the primary olfactory area of the cerebral cortex, which is in the anterior region of the parietal lobe, near the juncture of the parietal and temporal lobe, right posterior to the frontal cortex. If we look at a little bit more detailed schematic here, what we see is we see the pathway heading from the olfactory receptor to cranial nerve 1, from cranial nerve 1 heading into the thalamus, and then heading out into the various regions of the cerebral cortex. In this case here, heading out into the prefrontal cortex, leading to perception of the distinct odorant. So let's take a look at what taste buds are going to be dealing with. In general, we have three distinct types of taste buds. The three distinct types of taste buds are known as the valate, also known as the circumvallate, the fusiform, and the folate. Each one of these will be found in a distinct region of the papillae of the tongue. Within the taste bud, we're going to have three distinct classes of cells that we'll see. We have referred to as the support cells. These support cells act just like the support cells for the olfactory epithelium, which means they're going to produce a mucus. This mucus is there to trap chemicals within the gustatory receptors. And then we have what are referred to as basal cells. The basal cells act as a rigid support in order to make sure that the taste buds keep their shape. So if we look at the tongue or the lingua, we have distinct regions within there. Within the distinct regions, we're going to have distinct areas where we will find the various types of taste buds. Most of these taste buds will be found on the fusiform papillae, the filiform papillae, the folate papillae, and the volate papillae. If we look at the distinct types of gustatory receptor cells, based off of the shape of the taste buds, what we see is we see a clustering of chemoreceptor specialized cells lining the various pits or folds of the taste buds. In this region, we will get trapping of distinct chemicals. These distinct chemicals will then interact with the taste bud itself, triggering the taste bud to send an action potential based off of which types of taste bud receptor cells within the gustatory complex reach action potential. You do not get a distinct taste based off of a single taste receptor. What ends up happening is that within the taste receptor, the various gustatory cells will be excited or inhibited based on the type of chemicals found within the food sources. There's one type of food source that will cause these gustatory receptors to fire all at once. That one type of food source is what we usually refer to as the spicy foods. Within those spicy foods, the chemical that's going to cause this issue is what's known as capsaicin. We will talk about this a little bit later on. If we're to look at the pathway that's occurring, we have three distinct cranial nerves that will have terminals originating from three distinct regions of the tongue. The three distinct regions are going to be in the anterior, in the posterior regions of the tongue, where we have the facial nerve, the glossopharyngeal nerve, and the vagus nerve, all transmitting sensory information. These nerves then synapse on interneurons within the gustatory nucleus, where we have information then transmitted into the red nucleus of the thalamus, as well as into the arcuate, the ventral, posterior, medial, and lateral hypothalamic nuclei. From there, the information then gets passed to the insula and to what's known as the primary somatosensory cortex of the parietal lobe. If you notice, the nuclei of the hypothalamus and the nuclei of the thalamus are involved with the neuroendocrine response to feeding. What we don't have is we don't have a topographic orientation to taste, and we do not have a topographic orientation to smell. The reason why we do not have a topographic orientation to taste is because of the population coding event. We used to talk about taste as having 5: sweet, sour, etc. However, what we found is that the interpretation of data from a study in 1919 was inaccurately transcribed from German to English. When we went back to the original data, we found that we actually have taste for all sensations coming from everywhere within the mouth, in particular along the tongue. The greatest density of gustatory receptor cells happen to be for those that respond to fructose and to sodium chloride, table salt, which is why we have a greater affinity for food that have large amounts of table salt and large amounts of fructose in them. So let's take a look at these pathways again. Here is the olfactory pathway once again. But what happens when we start talking about consumption of food through the mouth? Taste, when we chew, we end up causing chemicals to become volatile, coming away from the food particles themselves. As those chemicals come away from the food particles themselves, we then exhale. The exhalation of those chemicals cause those chemicals to interact with the olfactory bulb, the olfactory epithelium, at which point in time we then get a total sensory information of both the taste as well as the texture as well as the smell of the food. This is why when you have a cold, foods not only smell very dull but taste very dull. It has to do with the interaction of the afferent pathways coming from the taste buds with those afferent pathways coming from the olfactory epithelium within the hypothalamus and within the thalamus, leading out to those integratory areas in the insula and in the parietal lobe, causing pathway excitation within the frontal lobe, leading to a full sense of taste and smell. You can see this schematic in a little bit better detail by following the link either in the video clips online through Blackboard or by pasting this video link into your browser.